SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટ ના માધ્યમથી જોડતી કડી એટલે સંધાન.
Hello, good afternoon viewers. Today we are here to talk on a topic which is related to the two surface appendages found in the bacterial cell. We are going to consider the structure and function of the two important appendages, namely flagella and pili. Myself is Sneha Pandya from MD Science Institute, and with me is Piyush Pandya from Gujarat Arts and Science College. And today we will try to give you the information. Primary information will be covered by me and the detailed information will be covered by Piyush. So let us start. The first slide, as you can see, this shows the typical prokaryotic bacterial cell structure. As all of us know very well that prokaryotic cellular organization is the simplest cellular organization found in the biological systems, and various structures have been displayed in this prokaryotic bacterial cell. We are going to highlight the two important structures which we know them as the surface appendages. As you can see over here, the one which is shown singly and which is a long wavy structure is the flagellum, shown brown in color, and you can see the presence of numerous shorter structures present all over the cell surface, and they are pili. The same has been shown here in the next photograph, where you can see the presence of number of flagella, which are long hair-like wavy structures present at the pole of the cell, whereas the smaller structures, fimbriae, as have been mentioned over here, they are present all over the surface. At this stage, I would like to say that the two terms pili and fimbriae, they are synonymously used with certain exceptions, where some special type of pili which are involved in some other important function, for them this term is not applicable. Generally this is applicable for the somatic pili which are there and which are responsible for giving the property of adherence to the bacterial cell. In the next slide you can see, now let us have the basic information about the flagella first. As it has been mentioned, the singular is flagellum, whereas the plural is flagella. These are the long wavy ape-like structures, and chemically they are made up of a protein which is flagellin, which is known as flagellin. We know that flagella are the locomotory organs of the bacteria, and they are responsible for the movement of bacteria. Flagella can rotate either in clockwise or anti-clockwise direction, and as a result of that the movement of the bacteria takes place. As it is known that the bacterial movement mostly is observed in aquatic environment, that is in presence of water, where relatively low viscosity is present, and bacteria, they show movement in response to the stimuli. The movement which is there as a response to some stimulus is described as tactic movement, and depending upon the stimulus, for example, if it is light as a stimulus, then the movement is described as phototaxis. If it is some chemical, it is chemotaxis, and if it is magnet, it is magnetotaxis, and so on. So in short, bacteria, they move or they show movement in response to the stimuli. In the next one, we have given the general information about the pili. Plural is pili, singular is pilus, and it is mostly found in gram-negative bacteria, and these are comparatively shorter and straighter structures than bacteria. They are tubular and they are also made up of a protein which is known as Pili. Now in the next slide, as has already been said, these are comparatively shorter, straighter, and they are sticky and bristle projections present all over the cell surface, and these are basically there to give the property of adherence to the bacterial cell. With the help of the pili, bacteria, they can adhere to another bacterial cells. They can also have adherence with their host cells. This is very important in case of pathogenic organisms, and they can also give the property of adherence to the bacteria to their substrates in the environment. And exceptionally, there are some special types of the pili which are also involved in the transfer of the genetic material from one cell to another. In the next slide, as I have already told you, the pili which are basically there for attachment, these are also known as attachment pili or somatic pili, and they are there to give the property of adherence, whereas the other type of the pili, special type of the pili, these are known as sex pili, and they help the transfer of the DNA from one bacterial cell to another during conjugation. How the event takes place, that we are going to cover later on in detail. Now let us see about the arrangement of the flagella. We know that the bacteria which are flagellated, that is the bacteria which are having presence of flagella, the development and the arrangement of the flagella on the different types of the bacteria is also different, and it is the particular or it is specific for the specific species. And the development and distribution of flagella on the bacterial cell surface has been considered as one of the important criterion for the classification of the organisms. Now let us see how the arrangement of the flagella is there on the bacterial cell. These are described as the monotrichous if the single flagellum is found. They are lophotrichous if the tuft, that is the bunch of the flagella is found at end of the cell. It may be amphitrichous when the flagella are found to be present at both the ends, and it would be peritrichous when they are found all around the bacterial cell surface. So this is an important characteristic, important parameter or important point to be noted. Here in this figure you can see the long wavy hair-like structures, flagella, how they are there present at the particular position. In the first it is mono, single flagellum is there at one end. In the second one there is a tuft of flagellum, bunch of flagella present at one pole. In the third one a single flagellum is present on both the poles, both the ends of the cell, and in the last figure it is the flagella, number of flagella are present all over the cell surface. Here you should note that in the first three figures, mono, lopho and amphitrichous, the flagella are present at either of the poles of the cell. They develop and they are present there at the end of the cell, at one of the ends of the cell. In case of the fourth figure, the flagellation is there found all over the surface, that is the lateral flagellation is there along with the polar flagellation, and as a result of that finally it is the peritrichous flagellation where large number of flagella are found to be present all over the cell surface. The same here, the color figures are there showing the different patterns of the arrangement for the flagella. In the next one, this is the SEMs, scanning electron micrograph, showing the various patterns of the flagellation. Here I would like to say that this type of the photography. It is the false colored type of the photography. You should not take this as the colored microbial cells. These are not colored microbial cells. This is the simple SEM showing the various patterns of the flagellation. And you should also be knowing that while observing the movement under the microscope, ordinary microscope that we use in the laboratory, we can see the movement of the bacteria, but we can't see the flagella even though they are there. And that is due to the extremely small sized, that is the extremely small diameter of the flagella, which falls below the limit of the resolution of the light microscope. And so flagella we cannot see, even though we are able to see the movement. And to see the flagella, we know that the special type of the staining techniques are required, and in then we can see the flagella after staining them with the suitable technique. In the next one, we are going to consider the different the detailed structure of the flagella, where here you can see the description is there about the different regions of the flagella, and all the details will be given to you by Piyush. So please.
Namaskar. Good afternoon, everybody. The background is already has been laid, and now let us talk in detail about the structure of the bacterial flagella. As already known to most of the students, the flagellar body itself can be divided into three different portions, starting with the basal body and then the hook and the filament. What we usually identify as the flagellum in diagrams and in figures and in photographs is actually only the third and external part of the flagellar structure. The three-phased structure contains the basal body, as I said, and then the hook and then the filament. What we see external to the cell is only the filament part, and that too, as Sneha has already mentioned, can be observed only if it is properly stained or has been given some other treatment so as to increase its width. Otherwise, in its natural diameter, the flagella or flagellum rather is not possible to be visualized using an ordinary light microscope. As the student fraternity must have always noticed, they would have seen the motile bacteria moving here and there quite often under their high power objective using the hanging drop preparation. But have you ever wondered why the flagella cannot be observed? The reason is known to you, students. The diameter of the flagella is so restrictive to the limits of light microscope that it cannot be observed. Anyway, that is the external part. The internal part, as I said, is known as the basal body, and the basal body and the filament, the external part, are connected with each other through what is known as a hook that acts as a mediator between the two. As this slide shows, the first part, the helical filament, is, as I said, what is observed externally. It is a long region and constitutes almost 90% of the total length of the flagellar structure. The rest 10% is constituted by the hook and then the inner basal body. The filament part of the flagella is made up of a protein which is called flagellin protein, and as we will see later in a video which we have, the flagella has a tendency of growing at the tip. That is, when the flagellar growth takes place, the flagellin molecules, individual flagellin units, are always added into the growing filament structure such that the latest body goes and places itself at the tip of the flagellum. So when you look at the tip, that is the part which is synthesized at the latest. We'll look at that video later on, not at the moment. Now these flagellin bodies are constituting into a single filament structure known as flagellum or the filament. Then, as we have already talked, the next is the hook, which provides the link between the filament and the basal body, and the part of the flagellar structure which is probably the most important for the movement of the flagellum and thus that of the cell is the basal body. The basal body, as you can read in the slide, is the terminal portion of the flagellum which falls inside the cell wall and cytoplasmic membrane, and it fixes the flagellum to the cell wall and the cell membrane. There is a central rod surrounding which there are certain discs present. Now the number of the discs is always dependent upon the kind of bacterial cell. If it is gram positive cell, there are only 2 discs, and if it is gram negative cell, there are 4 discs. We will be elaborating upon this looking at this diagram. As this diagram shows you, the structure on your left is that of a flagellar body of gram negative organism. You can see there are 4 different disc-like structures observed surrounding a central rod, and then there is a hook in between, and external to everything else is the filament. Now look on your right hand side, the structure of flagella in case of a gram positive cell. You can see what is conspicuous by absence are those 2 external discs which are seen in the basal body structure of gram negative organisms. The reason why gram negative organisms have 4 discs and gram positive organisms have only 2 is very interesting. The lower 2 discs, which are observed both in gram negative as well as gram positive organisms, are called the S and the M disc or S and the M rings. These are the rings which are necessary for the rotary movement of the flagellar structure. The upper 2 rings, which are found only in gram negative organisms, are known as the lipopolysaccharide and peptidoglycan rings. These names are derived after the fact that they are lying close to the peptidoglycan and lipopolysaccharide structure of the cell wall of gram negative organisms. Gram positive organisms do not contain these rings. Now let us elaborate upon the reason. Look at the comparative structure of gram negative and gram positive organisms. You will immediately find the difference, and I believe and most sincerely hope that the students should be aware of the fact that cell wall of gram positive organisms contains a large quantity and a very well fabricated layer of structure known as peptidoglycan. In case of gram positive organisms, peptidoglycan structure is present as a thick layer, very well fabricated layer, and it provides the mechanical support to the central rod of the basal body. In case of gram negative organisms, however, the cell wall contains very little amount of peptidoglycan, and instead it contains another layer known as lipopolysaccharide layer. Because of little quantity of peptidoglycan in the cell wall of gram negative organisms, the mechanical strength cannot be provided to the central rod of the basal body by the cell wall structure, and therefore nature has provided gram negative organisms with 2 additional discs, 2 additional rings, the peptidoglycan ring and the lipopolysaccharide ring, the L and the P ring respectively, which provide them the mechanical support. Let us have one more diagram which once again depicts the structure of the basal body and the hook and then the filament. We still have one more slide where the comparison between gram negative and gram positive organisms cell wall becomes much more clear. If you look at the upper diagram over here, you can see this is a thick and very well fabricated peptidoglycan layer. This is a cell wall of gram-positive organisms. These two, the M ring and the S ring, and then the central rod which is supported by these peptidoglycan layer, and therefore only two discs which are needed for the rotary movement are there, and then the rest is done by the peptidoglycan, that is providing the central rod with the mechanical support. Now look at the cell wall of gram-negative organisms. Here we have a comparatively thin peptidoglycan layer. You can check the difference. These gram-positive organisms and these gram-negative organisms. You can check the difference. So comparatively thin and less fabricated peptidoglycan of gram-negative organisms cell wall cannot provide the strength which may be needed to support the rod, and therefore they are provided with two more discs. You can see one here which is called the lipopolysaccharide disc or ring, and this is the one which is called the peptidoglycan ring. These two collectively provide the mechanical strength. What is very important to realize, students, is that these two, the S and the M, S for surface of the membrane, M for membrane, these two discs are basically needed for the rotary movement of the flagella. So in those organisms, gram-negative organisms, where these two additional discs are there, their role is only very mechanical, of providing the strength to the central rod. Fine. This is one more slide which shows you the same thing: the cell wall, the cell membrane, the two discs and the hook. One more diagram showing the same thing. You can see this is a cell wall of gram-negative organisms, and therefore there are two additional discs, the lipopolysaccharide ring and the peptidoglycan ring. I believe with series of these diagrams of different variety, I have been able to make it clear to the students why gram-negative organisms have 4 discs in their basal body and why gram-positive organisms may have only 2. This is a figure I want all of you to look very closely, because this figure is only going to be converted into a live diagram where you would be able to see the rotary movement of the discs, the S and the M rings, and this is how the movement passes across this hook to the filament. Fine. So please remember this. Now let us first of all talk about the motility of bacterial cells. How are they able to move? As Sneha has already mentioned, we have to understand that in most cases the bacterial cells which move with help of flagella, and most move with help of flagella only, are showing the capacity to move only in the aquatic environments, in the liquid medium, where the viscosity is not very high. With increasing viscosity, the capacity of the bacterial cells to move decreases. We have to remember that the bacterial cells are not moving, they rather swim in a liquid environment. The other property which finds a mention in this slide is that of antigenicity. The bacterial cell, the flagellum of the bacterial cell, is also providing the cell with the capacity to act as an antigen. The H or the flagellar antigen is always situated in the proteins of the flagellar structure. Anyway, because it is not to be elaborated in this particular discussion, we will not be talking in detail about that. Now when we talk about the ability of the bacterial cell to move, we have to remember that the bacterial cells move with help of the flagella. Now how do the bacterial cells move with help of the flagellum? The flagellum, as has been repeatedly told by both of us, is a very delicate, very fragile and very thin structure which cannot be used by the bacteria to stroke the inertia of the water bodies in which they have to swim. Like when we or any animal, we swim in the water, what we do is we stroke the water with our hands and legs, and then the inertia of the water, because it is stroked against by our appendages, gives our body the forward motion. This kind of mechanism cannot work in case of bacteria. The reason is very simple. The flagella itself, the flagella themselves are so delicate. If they are applied to stroke the water body, what will happen is that they would be broken because of their fragility, because of their delicacy. So what happens in case of flagellar motility is that the flagella rotate, and as the laws of physics always tell us, the rotary movement is always much more penetrative than the straight motion. So because of the rotary movement, the bacterial flagellum can pierce the water inertia, and thus it can make the bacterial swim in a water body, in a liquid body. This rotary movement of the bacterial flagellum is generated from the base, from the basal body. The lowest disc, as we have already mentioned, the S and the M ring rotate around the axis by a mechanism which goes beyond the discussion of this particular session. Sometimes, if the time permits, we will come again and tell you about the exact mechanism. At the moment, it is only enough to remember that there is a specific force generated within the bacterial cell known as the proton motive force. Because of the proton motive force, the rotary movement of the S and the M discs is generated, and once the S and the M rings are rotating, this rotary movement passes across the rod to the hook, and via the hook it goes to the filament. The filament rotates, strokes the water body, and that is how the bacterial cell moves. And this shows how a bacterial cell can move, and we have to remember that movement of the bacterial cell is actually the displacement of the bacterial cell. It has to move from one place to another. You often find the bacterial cells suspended in a liquid medium, moving here and there and stabilizing themselves to a specific point, which is called the Brownian movement. This may not be confused with the flagellar movement, which definitely involves the displacement of the bacterial cells. This slide shows how a bacterium with peritrichous flagellation can move. You can see the peritrichous flagella form a bundle, and the entire bundle rotates in a particular movement, and when the bacterial cell has to change the movement, the bundle of the peritrichous flagella is unfolded. The bacterial cell undergoes a tumble. This tumble changes the direction. The bacterial cell once again prepares a bundle of the flagella and then starts rotating them in another direction so that it can move in the desired direction. And this shows how a bundle of flagella moves. This video actually shows the entire movement of the bacterial flagella. You can see the lowest is the S ring, the lowest is the M ring, then the S ring. Then, because this is a structure depicting the flagellar basal body of a gram-negative bacterial cell, you can also see the T and L ring, but you can see these two are not rotating. The rotation is observed only with reference to the M ring and the S ring. The L as well as the P rings, which are involved in providing the entire body with the mechanical support, are not rotating. This movement, this rotary movement of the M and the S discs passes across what is known as the rod, central rod. It moves to the hook, and via the hook it transfers to the filament. You can see the filament is rotating around a specific axis. We have to remember that at the moment this filament, in this diagram, in this film, the filament was shown moving in clockwise direction. When the bacterial cell has to change the direction, the same filament can stop and also start moving in anti-clockwise direction. That will help the bacterial cell change the direction. At the moment, that should be enough about the bacterial flagella. Now let us talk about the pili, the another variety of the bacterial surface appendages. Bacteria, as you have already been told, bacteria have two types of surface appendages, the flagella and the pili. The flagella are lengthy, wavy and whiplash-like structures which are involved in the locomotion, and also they provide the bacterial cell with antigenicity. We have already made a mention to that, the flagellar or the H antigen. The pili, on the other hand, are comparatively shorter structures. They are straighter and very mucoid. Let us now talk about the pili. They are the short appendages which are made up of proteins. They are naturally smaller in size than the flagella. The basic function of pili is that of adherence. We will talk about that later on. They are also used in the process of conjugation, where they are involved in providing a bridge between two conjugating bacterial cells through which the exchange of genetic material takes place. They also help the bacterial cell increase their buoyancy by raising their flotation rate. This structure, this diagram shows the structure of the bacterial pili. You can see at the base we have the cytoplasmic membrane, then the peptidoglycan structure, and at the top we have the cell wall lipopolysaccharide structure, through which we can see the pilus, a single pilus molecule piercing out as an appendage. Like flagella are made up of a protein called flagellin, pili are made using a protein known as pilin. Needless to tell you, the pili is the plural and they can be a single pilus, singular is pilus. The basic function, as we have already said, is adherence of the bacterial cell. They are responsible for the formation of the biofilms on several substrates, and they also add to the pathogenesis of several organisms, pathogenic organisms which have them, by their ability to adhere the bacterial cells to the respective surfaces where they have to go and attach for pathogenesis to establish. This is a photograph, a scanning electron micrograph actually, which shows a bacterial cell containing numerous pili. It is very important to remember that unlike flagella, the pili are always present in a large number all around the bacterial cell. Flagella, as we have already talked, can be mono or amphi or lopho or peritrichous kind of arrangement. Pili don't have that kind of differences in arrangement. If the bacterial cell has pili, if a pileated cell is there, the cell is always surrounded all over by the pili. An exception to this is that of the sex pilus, about which we will talk later on. This is one more false color electron micrograph showing a bacterial cell with several pili. For a change, this is a transmission electron micrograph and not a scanning electron micrograph as the previous photographs were. This is also a photograph where you can see the role of pili in conjugation. Now this is a reference I was making earlier on. The pili are basically of two types: the somatic pili, which are found all around the cell, and the function of these somatic pili is that of adherence. But then we also have a special variety of pilus which is known as a sex pilus. This sex pilus is also called the fertility pilus or the F pilus. What is important to remember is that a single bacterial cell, if it has a pilus, sex pilus, will have only a single sex pilus at one of the poles of the cell structure. You can see here. This is one more example and a very famous photograph of two bacterial cells which are connected with each other by help of a sex pilus. This is in the process of conjugation. The cell on the left is the one which is pileated, which contains a sex pilus, and it has extended its sex pilus towards the one at the right hand corner, and the two have been conjugated, the two have been united with help of that sex pilus. Once the two cells are connected with help of the single sex pilus, the pilus forms a bridge between the two, across which the genetic material from the donor cell transfers into the recipient cell. This is once again a very famous photograph showing the role of pili in conjugation. This is once again a false color photograph, transmission electron micrograph. You can see here different bacterial cells being connected with each other through the sex pilus. These are certain websites which we would like you to visit at the time where you are studying, and whenever you find time to go and visit your net facility. Please visit all these sites. I am keeping this slide for a little longer than I would have, so that you can copy the addresses down. These are the sites where you can see excellent photographs and animations and even videos regarding the structures of flagella and pili, and also how the flagella move. The videos as well as the pictures that we have incorporated in this presentation were all collected from these websites only. So we also acknowledge and thank the creators of these websites. We have with us two interesting videos which we would like you to watch. Please do. Primarily, this video shows phase contrast photovideography rather, which shows the bacterial cells moving with what appears as a single flagellum is actually a bundle of flagella. Please note that this is not a single flagellum. Now look at this video very carefully. This is an animation actually. Look. This part is the hook. This is the filament, as the label itself suggests. And as we move down, you can see all four discs, the basal body structure. The basal body, you can see, has started rotating the rings as depicted here. As I had already made a reference to the proton motive force, though we don't go into the detailed biochemical processing of this, just look how the proton motive force makes the whole thing rotate. Look carefully. We have decided to incorporate this video in our presentation because what a lecture of 1 hour cannot tell you, this video will be able to tell you. With help of the proton stroke, you can see the rotary movement begins. The protons are thrown out, and then the reverse. Power stroke 2. Look how carefully nature has designed the motor of tiny bacteria. This is a motor no individual can ever design. Now look, this is the inner membrane, this is a peptidoglycan structure as you already know, and how the flagellum is synthesized. The synthesis of the entire bacterium flagellum is now being presented before you. The inner core is in the process of being synthesized. You can see it coming up. These are all protein molecules. There are certain structures to which we have not made a reference, for the obvious reason that we have prepared this particular presentation for the students of undergraduate, where all these details are not necessary. Now look, the rod, the central rod is being synthesized. In the process, it has been surrounded by those discs, the L and the P rings, so that the rod structure is given the mechanical strength. And now we will start the synthesis of the hook. Look, the hook is in the process of being synthesized. The hook protein units are being added, and once the hook is, you can see the hook cap has been removed. What now will happen is the synthesis of the filament. I told you in the beginning that the flagellum grows at the tip. That is the units of the flagella. The flagellin units are always, the fresh unit is always added at the top. This video will show you exactly how. Look, every single unit travels all the way right to the top up to the cap, and this cap will remain for as long as the complete length of the hook is synthesized.
Filament.
Of the filament, I am sorry.
Very nice. ये गई. बीजू करते हैं.
Please, we have one more film to show which contains a little part of the one which you have seen, but that is also quite interesting. This film shows only the synthesis of the entire flagellum, but at a faster rate you would be able to appreciate the entire process
Quickly.
So it is only the part of the earlier film, but moving at a faster rate, and it shows you the part in which I believe the undergraduate students would be more interested.
Capture theory.
You can see the hook cap is still there. Now the hook cap will go.
Yes.
The hook is almost synthesized, the cap goes away.
Flagellum.
Now the filament. There will come out a filament cap. You can see this cap will remain for as long as the entire length of the filament is synthesized, and then the filament cap will also go, and then the bacterial cell will start moving. By visiting the websites that we have mentioned, you will find such videos which will make your understanding related to the structures and functions of various microbial organelles very clear. So as we come to the conclusion of our discourse, here is a suggestion from both of us to the students that you have to learn from these tiny structures of bacterial cells. Your teachers would have often told you to learn from the bacteria, but we also suggest to learn from the internal structures of the bacteria, the external appendages of the bacterial cells. From flagella, what you can learn is that make a selfless attempt to move the society. You have to always contribute to help the society move in the right direction, like a flagellum does. It always helps the bacterial cell to move in the right direction. The flagellum, have you ever heard of bacterium moving in the wrong direction because its flagellum did not function properly? No. So in a similar manner, a good citizen, a good human citizen is the one which will help the society move in the right direction. In a similar manner, from Pili also you have to learn to adhere to your goal very firmly. You are at a stage of your career where you have to set your goal and then adhere very firmly to that goal, so that, like Swami Vivekananda said, this is the year of 150th birth anniversary of Vivekananda ji, and thus I cannot refrain from reminding you of his famous discourse, Utho Jago ane Dhyay Prapti Sudhi Mandya Raho, and this is what Pili teaches us. So, student friends, as we come to the end of the discourse today, if you have any questions, please ask. Otherwise, from both of us, with very best wishes for a future very bright and brilliant, thank you very much. If you have any question, please ask.
SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.